Kathy, so much for the invitation. And thank you all for staying on a Friday evening for the last talk. So thank you very much. So today I'm going to talk a little bit about how gardens can help with conservation and work with plants both in situ and ex situ. So anybody who's not familiar with these terms, so in situ is in the wild. This is where we're actually doing restoration, monitoring, augmentation, reintroduction, all of these different things that I'll touch on in the wild. So we're actually going to those populations and affecting and working and monitoring those populations in the wild. Ex situ is when we pull them out of the wild because their population is going to get destroyed for some reason. Or we're trying to safeguard them because they could potentially be facing some form of loss. So we do this with seed banking, we do it with live plants where we actually grow them out. We have a safeguarding nursery that I'll talk to you about in just a second. So, since we're at Emory, and just to explain a little bit about what our mission is in the Conservation and Research Department, we work to promote innovative strategies and partnerships to conserve imperiled plants and natural communities. So while we are a botanical garden and while we work with primarily very rare, endangered, threatened species, we are really interested in making sure that we conserve the whole system. So a rare plant, so a rare orchid, let's say, that occurs here and right outside of Atlanta, we're really interested in that particular plant and the uniqueness of that plant, but if we don't make sure that we work with the whole system, we are actually missing something. So in our department, we try to work at that whole picture scale. We do this work and carry out our mission through research, collections management, where we actually hold collections at the Botanic Garden, share collections with other Botanic Gardens, other researchers. We work with focusing on priority habitats, something that I'll talk to you about. And we do a lot of educational outreach to try to explain to people why they need to save plants, why they're so important, and ecosystems. And finally, no plant can be saved alone. We do this work through very, very interdisciplinary collaborative partnerships that we work across the region and internationally to save these plants. It's really important to us that we are making these meaningful connections and partnerships to make a difference in these plants' lives. So we have several strategic priorities. We work with prioritize species that are carnivorous, and we also work with a lot of orchids. They're very cool indicator plants. They tell us a lot about an ecosystem. They're also pretty charismatic, as you can imagine. Carnivorous plants, it's a nice story there. We also work with globally ranked G1 and G2 species. This is a ranking that, as is the IUCN red listing, this is a ranking to help us understand how rare a plant is in a particular habitat, what the threats are, how many there are, how big the populations, genetic variability, these sorts of things. So we work with what are basically considered the most rare, the most critically endangered, and that's also a G1. And because these are different, so NatureServe and IUCN, they have slightly different criteria, but we work with the most endangered, most threatened species. We conduct we conduct our research to help us really inform actual active management on the land and in situ efforts, so all of those wild efforts. It's really important to us that our research and our data has a meaningful impact to the plants and to the land manager, so they can utilize that information and then actually make a difference with it, with how they deal with these plants. And because we happen to live in one of the most biodiverse hotspots in the world, the Southeast is a very large biodiversity hotspot. We prioritize habitats that are very unique to the system, including the bogs, wet savannas, fens, these sorts of areas. Seepage slopes, longleaf pine ecosystems, we work in coastal plains. So we work sort of all across the Southeast. We also do work internationally, but these are sort of, we actually focus international work around wetlands and bog sort of habitats. This is a picture of our totally fantastic, wonderful team. And I have most of our team here today, and the only person who's missing is Dr. Jeff Stevens, who just joined us after this picture was taken. But this is a picture of us down at the E.O. Wilson Biophilia Center. So we do a lot of work down there. We have a very large restoration site that I'll touch on for just a moment. And we had our interns that came with us down there. We have a really rigorous internship program where we had 8 interns this past summer. And we currently have 4 interns this fall. So the main sort of over the big boxes that I like to sort of talk about is we have research, and this guides, so the research that we do in our department, this guides how we work on restoration, monitoring, and our outreach. So our research involves molecular work and also both in situ, ex situ, looking at health of plants in some cases with some of the work that we do down in Fakahatchee, looking at reintroduction rates, survivalship, these sorts of things. And we do large-scale restoration and small-scale restoration. So we work in the North Georgia bogs, and these are fairly small, 1-2 acres, if that, sometimes wetlands and bogs, all the way down to our coastal dune lake project that is 320 acres down in the panhandle of Florida. This is a map showing the 24 sites that we work in with just carnivorous plants. We have maps like this that are for all of our other species, but this is just the carnivorous plant work that we do. So we work across 7 different states, and we have just begun working in Puerto Rico. For our orchid work, it looks similar to this, except it goes further down south into Florida and includes Cuba, where we are working with local partners down there. I'm just going to talk about a couple of fun projects that we have done. So given that there have been many talks about importance of pollinators, and Asclepius is one of the projects that we've worked on. So we go out, this is actually at Panola Mountain State Park. We've gone out, we collect seeds, we will propagate those seeds, we will outplant the seeds and help with prairie restoration, because we've had a lot of loss of these species due to habitat conversion, overuse of pesticides, and herbicides, and land management practices that have reduced fire, reduced a lot of the natural systems. Additionally, we've worked with the white fringeless orchid, so Placantha imperiled. It's this beautiful orchid here. This site is not very far from where we are right now. It's about 45 minutes away. We have gone out and looked for field surveys. We have a lot of herbarium samples that are historical samples that we use to understand where populations used to be. Then we can go and try to ground truth that to see, are those species still there? Especially for really rare species. We may have an herbarium sample that is from 100 years ago, 50 years ago, sometimes sadly 10 or 15 years ago, and we want to see, are those plants still out there? So we'll use that information, go back out, try to find the plants. Sadly, a lot of times we don't find them always. That's the case in our South Florida work. So this is Matt Richards, and he's been doing work down in the Fakahatchee Strand. There are 4 species that have been extirpated from that region due to poaching and habitat loss, but we have herbarium records that indicate that they had been there historically. So with this project, it's very similar to others. We grow out, we'll conduct surveys to really look for look for the plants if we can find them. In some cases, we end up having to do hand pollination, depending on how healthy the system is, if we still see pollinators around. And then we will collect seeds. We have a micropropagation lab at the garden where we're able to grow these microscopic seeds out, transfer them to our nursery beds, and eventually get them back out into the wild once we have restored that habitat. We also do this up in the North Georgia mountain bogs. These are some of the beauties that we work on. We managed 20 bogs collectively with the Georgia Plant Conservation Alliance, and we do habitat restoration, invasive species control, and then augmentation of populations. That's some of the orchid research that we do. So this is actually Matt down in the Fakahatchee Strand. He is doing some work on the cigar orchid. which there were 19 individuals of this plant left when we were brought in to help with it. They've been able to pollinate these and then grow them out, and we've been able to have success with some of this outplanting. We additionally do a lot of documentation of orchids in the Southeast. We do work with monitoring and seed collection in Cuba. We work down in Ecuador, we've done some molecular systematics work down there with orchids, and we also are currently conducting barcoding of our collection. So we have over 2,000 orchids, 2,000 orchid taxa within our collection at ABG. And Dr. Lauren Easterman is actually the one who's conducting this research. So she is taking samples from all of these plants, and then we're doing barcoding on them. In addition to that, we are also doing some deeper dives to look at some phylogenetic questions that we're interested in with some of the very complex groups that we're interested in researching. And finally, I'll talk to you about sort of a big project that looks at historical changes and fire management, what we were doing down in Deer Lake. And so this is in between Panama City and Destin, where the Deer Lake State Park occurs. This is our large 320-acre restoration project. Fire has been suppressed for the last 100 years, and with that you get this overgrowth of tai or Cliftonia. And what once had looked like this beautiful scene, this is what the system has been converted to. It's very dark and dense, there's very, very low diversity. This is one of the areas in the Southeast that would have extremely high biodiversity. So what we're doing is we're converting this into this. This is actually our first pilot project plot, so this is the actual result that we started back in 2014. And so this is this year's photo from that same area. And this project is really long. It goes until 2023, which is really exciting. What we do is we actually clear this Cliftonia with these very large, these feller bunchers. All right, this is the feller buncher right there. So we remove that, we mulch it, and then we get it off-site. Because these are nutrient-poor ecosystems. They do not want a lot of extra added organic matter to them. If you have that added, they will not, the plants will not do well, especially things like our pitcher plants that we're really interested in. So we remove all of this biomass. That is obviously an immense amount of material that we remove off-site. Luckily, we're able to sell it to the paper company, and they're able to make paper with it. And then we reintroduce fire, because fire naturally would keep this system more open and be able to really allow for that diversity to happen. So we utilize prescribed fire as our management tool. And then we come in and we have done seed collection from this site and Eglin Air Force Base, which is right next door. And we then augment and reintroduce plants that had been in these areas. To date, we've replanted over 4,000 species. There are a number of Sarracenia, so we have our Leucophilus, our white top pitcher plant, and our parrot or Citacina pitcher plants. We have numerous of our Drosera, so our sundews. We also have some orchids. That we have been able to outplant and augment this population so that it's actually going to get back to functioning hydrology. We do this through our micropropagation lab, so this is just a picture of where all that magic happened. This is normal times. When you visit ABG, you can actually see this right now. We're building our Southeastern Center for Conservation, where we're going to house a new micropropagation lab and a new molecular lab. That will be opening this next summer, so we're very excited. But normally you can walk by and actually see all of these plants. There's well over 10,000 plants within those little tiny vials, all stacked up together, and you can see here's some of the little baby Sarracenia, which are very cute. In that site, we then do long-term monitoring. So these sites that we're working on, it's a long-term plan where we're going in and really looking to see how the effects of restoration, what has happened and what did we see with recruitment. And finally, we were very excited. We just got a few more grants that we'll be able to do more monitoring work there, where we're actually going to start doing a full floristic inventory, which is great. More pollinator studies and also some amphibian monitoring, because these areas have been historically very important for amphibians, including the flatwood salamander, which is a federally listed species. And the last thing we do is our pollinator, and I'm sorry about that picture being off, our pollinator restoration program. And this is where we actually come in and we install pollinator gardens that we've done around the Atlanta area. Our pollinator restoration coordinator, Melina Lozano-Duran, she has led this force to put in 7 of these different pollinator gardens around in the city. We hold STEM events at them. We do pollination studies that she's been conducting this past year with an intern at 2 of them. And we really promote the use of good pollinator plants and also the importance of pollinators in our community, as well as conducting these surveys that we're trying to just sort of start and expand some of that work. And we have a really active conservation training program where we get a great cohort of interns every summer, and then we have credit interns that come through every term, which is really exciting, and we really love being able to bring them in. And finally, we are setting up our Southeastern Conservation Seed Bank, where we will have seeds available for, there'll be from the Southern, we already have a large collection of Southeastern U.S. native orchid seeds, and these are all used for conservation and restoration, and we're expanding that to include more species and also the Caribbean. So with that, thank you.